Let me share with you your sample. For those who have submitted. Sekejap. Okay, so let's go through one by one the questions. Okay, the first question, it asks you, sekejap. Do you have any question just now? Like state, fasting state, and all the integrations? No, I can continue. Yes,
Yang proteolysis tu yang amino acid tu kan?
yes, yes.
Okay.
Okay, so for the first question, so again, as I mentioned just now, for essay questions, only chapter 7 and 14 would be there. Okay, but what I want you to look into this example is that, so this question asks you illustrate with suitable diagram the effect of substrate concentration on the reaction velocity of an enzyme. Okay, so I just put the marks I just simplify the marks for each question. Okay, but for your exams, it should be the total marks for each question is 25 marks. Okay, so if the question asks you illustrate with suitable diagram or explain with the aid of diagram, you need to draw the diagram on your own. Okay, you cannot, first thing, cannot copy paste from other places. Okay, you must draw on your own. If you have problem in attaching in the BEZ, you can email them separately to me. Okay, just write in the question number and your names. Okay, so first thing, you cannot copy paste, have to draw on your own. Okay, second thing is diagrams must be accompanied with explanation or the written form of it. So for this question, most of you only draw the diagram and leave that as it is. Now you cannot do that. You have to explain the diagram. Okay, so although the question asks you to illustrate, but it have to be accompanied by explanation. Okay, so for this example over here, 2 marks are actually allocated for the diagram itself. Okay, so the first mark is it is a correct graph. Okay, it should be the graph for effect of substrate concentration on reaction velocity. So if you draw wrong graph, like you draw pH graph ke, temperature graph ke, then it would be wrong. Okay, so the graph must be correct. So another element or marks would be allocated for your labeling of the graph. Okay, your x-axis, y-axis. Okay, and if you are labeling the maximum point or the Vmax. Okay, for this one I can give like increasing concentration. Okay, but you can write in maximum point or Vmax. Okay, so labeling must be correct. Okay, and you please use a full word. Okay, don't use like max point. Okay, please write in maximum point. Okay, and then same goes to rate of reaction, substrate concentration. Don't just write substrate cons, CO and C. Okay, please write it in full form. Okay, just now I've received some example. Okay, someone draw something like this. And then label. Okay, something like this lah. Okay, and then level Vmax. So this is wrong because Vmax is not beyond the graph. It has to be matching or aligned with the maximum point of the graph. So it has to be here. Here is wrong. Here is correct. Okay, the Vmax labeling or the maximum point labeling. Okay, and then what you need to do is explain the graph. Okay, so let's look into how by illustrating with the diagram can help you to explain. Okay, so to make it simpler, you can do it like this. Okay, so this is the diagram, for example, that you have drawn. Okay, again, make sure you draw it on your own. Okay, let's divide this graph into 3 sections for us to make it easier to explain. First section, second section, third section. Okay, so first describe based on what you see based on this section. Okay, so this is the answer scheme for the content. Okay, so this is point number 1, point number 2, this is point number 3, and until 5. Okay, so let's look into it. Okay, so the first, what you can see now, okay, in the first section is that the rate of reaction increase rapidly, okay, because the graph we see here is very steep. Okay, so you can write in the rate or velocity of reaction increases as the substrate concentration increase. Okay, so that's for point number 1. So what happens next? We can see at the second phase here. So as the reaction proceeds, okay, the high concentration of substrate, the enzymes will get saturated and the velocity will start to decrease. Or you can say the rate of reaction becomes slower. Okay. So then you can also mention, okay, the concentration. So this is increasing substrate concentration. So let's say this is 0, this is 10. Okay. So what is kept constant here is concentration of enzyme. So the concentration of enzyme is kept constant. Okay. Increase in the concentration of substrate. Sorry. Okay. This will eventually reaches maximum point. Okay. So this is the maximum point. So after this specific point, further increase in substrate do not cause an increase in the rate of reaction. So you can see substrate concentration is increasing in 6, 7, 8, 9, 10, for example. But rate of reaction will eventually leave a stagnant state or a plateau. Okay, so why does this happen? So you can explain. So once the maximum velocity is reached or rate of reaction is at the maximum point, so all of the enzyme molecules are converted to enzyme substrate complex. So this is the analogy here. Okay, so the yellow colored ones are enzymes, orange molecules are substrate. So eventually, if you see the number of enzymes are kept constant, but the substrate molecules keep increasing, and eventually all of the enzymes are occupied. You can either say things like this: enzyme molecules are converted to enzyme substrate complex, or you can say all of the active site of the enzymes are occupied by the substrate. That can also be accepted. Okay. So as for the introduction, you can write the definition of enzyme. For example, you can write the definition, what is enzyme substrate, okay, what is the product, or maybe you can write one sentence, what is the function of an enzyme, okay. And for conclusion, you can summarize all your points in 1 to 2 sentences. Okay, so I guess the common mistake for this question is just that everyone just draw and leave it like that. But always remember, in any question asking you to illustrate with diagram or draw, you still need to make sure the drawings are accompanied with written explanation. Okay, so that is for question number 1. Okay, so question number 2, what I want you to focus on is that this question actually has 2 parts, okay? 2 hints for you. So the question asks how glucose is metabolized to yield energy. So you need to explain how glucose is metabolized, and you need to show how this metabolism yield energy. Okay, so usually, so this is 10 marks is just oversimplified, but usually it will give you marks based on oh, ni. Okay, so what you can do, you can draw this flow without the chemical structure. Okay, you can actually draw this from glucose and until pyruvate. So marks will be given like this. Glucose 1 mark. Okay, glucose 1 mark. Glucose 6-phosphate, the product of it is 1 mark. And then the enzymes catalyzing this reaction, hexokinase 1 mark. If you add in ATP converted into ADP, you can still get marks here, and so on. So this is actually a very helpful question for you, so you can get marks just based on the writing the pathway, the flow. Okay, so this must be, if you want to include this diagram, it must be written by you. Cannot copy paste from other places. Okay, same goes to, okay, this is 1 mark, 1 mark, 1 mark, 1 mark. Okay. So what you can do, okay, later after you have explained, you can also write this in written form. You can say, the easiest way is to say it like this: glucose is converted to glucose 6-phosphate. This reaction is catalyzed by hexokinase. ATP is converted into ADP. Then you can proceed. Glucose 6-phosphate is converted to fructose 6-phosphate, catalyzed by this isomerase. Then so on. Fructose 6-phosphate converted to fructose 1,6-bisphosphate, catalyzed by this enzyme. Okay, and ATP is converted to ADP. Okay, so the extra tick here is in terms of the flow itself. After you have written how glucose is metabolized and include the changes in ATP, okay, it's good for you to write a summary of how this actually yields energy. Okay, meaning after you have written these reactions over here, including the ATP changes, okay, you can write one sentences concluding that, okay, it consumes 2 ATP, okay, this one, but it can also produce 2 molecules of ATP. Okay, you can also include the localization of glycolysis, which happens in the cytosol. Okay, you can, that's for the introduction. Okay, you can also include this content. Okay, so this is just to show you on how you will be marked. Okay, so remember, you can actually write in this diagram flow, but you still need to explain it. Okay, this is just for make it easy for you to remember. Okay, so what I can suggest you to do is you can create a mnemonics, okay, to remember the enzyme names or even the intermediates names. You can just Google mnemonics, enzymes, glycolysis, mnemonics, intermediates, glycolysis. Okay. So again, you have to, if you want to include diagram, draw it yourself. Okay. So I think this table would be helpful for you to remember the flow, okay, and to not miss out points. Okay, any question for question number 2? Okay, wait. I just remember some of you also included the Krebs cycle in this part, but for this specific question, it only asks you how glucose is metabolized to yield energy, so you should be focusing on glycolysis. Okay, no marks will be given if you write Krebs cycle in detail, but you can write in how pyruvate can eventually enter the Krebs cycle to produce more ATP. Okay, that can be given marks because it's related to yield energy, but the details of Krebs cycle, that we have no marks for that, for this particular question, because it's only asking you how glucose is metabolized to yield energy. Okay, sekarang kau apa? Okay. Let me share you another example. Okay. So this third question asks you explain ketogenesis and ketone bodies. So for this one, most of you makes mistakes that you don't actually explain the 4 steps in ketogenesis. Okay, for example, over here, okay, you can write in the introduction paragraph, for example, the definition of ketogenesis. Okay, what are the examples of ketone bodies? Okay, you can write in when ketogenesis takes place. Okay, what trigger ketogenesis? Okay, that's within the introduction point. Okay, but what you need to do is actually to explain ketogenesis in detail. Okay, what happened? I noticed that most of you, you spend a lot of time in explaining how ketogenesis developed. Okay, when you should be, but you don't really include the 4 steps in ketogenesis. Okay, and some of you makes mistake like ketone bodies is produced by gluconeogenesis. So again, gluconeogenesis is about producing glucose from non-carbohydrate sources, while ketogenesis is about producing ketone bodies. Okay, so let me show you the answer scheme for this one. So it's like this. So marks will be given when you write in the name of the reaction. So we have 4 types of reaction in ketogenesis. First, condensation. Second, condensation. Chain cleavage and reduction. Okay. So if you name, okay, first step is first condensation, then 1 mark. So what happens in first condensation is 2 acetyl-CoA combine to produce acetoacetate, then 1 mark. Okay. And then what are the enzyme names catalyzing the reaction? Thiolis, 1 mark. Okay, and so on. The name of the reaction, 1 mark. What happens in the reaction? Who combines or react with who to produce what? Okay, and then what are the enzyme names catalyzing the reaction? Okay, so this is if the question asks you to explain ketogenesis, then you have to go into details of this. Not only what trigger ketogenesis, that would be a good points in the introduction, but you have to go into detail step by step in ketogenesis. Okay, and you have to explain what are the example of ketone bodies. Okay, which is the 3 things: acetoacetate, okay, beta hydroxybutyrate, and acetone. Okay, any question for this third question?
Doktor,
Yeah,
ketogenesis ni reversible kan?
reversible.
Hmm.
Betul.
Okay.
So basically, most reactions that are catalyzed by enzymes are reversible. So not only in ketogenesis. Okay, if you
Okay, doktor.
remember in chapter 5, the characteristic of enzyme, usually the reactions are reversible. Okay. So can I continue with next question?
Boleh, Mida, boleh.
Okay. So the next question is basically on food biochemistry. Okay, so in this the practice that I've asked you, basically I asked you to write in all the biochemical functions. Okay, but usually in exams it's more specific. How many you should be writing in your answer. Okay, for example, it asks you discuss or explain 5 biochemical functions of what. Okay, but sometimes it's not that straightforward. Let me show you the food biochem notes. That's why I asked you previously to complete this table. Okay, it's this deficiency table. Okay, I have given an example for fat soluble vitamins. Okay, you can change the chemical names. You don't need to remember the chemical names, but try to find what are the sources for each vitamins and also minerals. Okay, where can you get these vitamins and minerals? Okay, so question may ask you based on a case study. For example, a patient who is experiencing certain conditions. Okay, so what maybe the question can ask you give 7 examples of vitamins, minerals, and other molecules that are required by the patient to recover from that disease or state. For example, okay, that's why you need to do this table. Okay, I have actually put the link in the lecture note. Okay, you can go to this link later on and read through them and just take out the information and put this in the table form. So that's better for you to revise. Okay, so questions on food biochemistry is usually straightforward. It may only ask you, okay, patients with this condition, how would you advise? Just like your assignment 2, kan, you have dietary suggestion for pregnant women ke, for children ke. So that is based on your knowledge of this, which is summarized in this table. Okay, so you really need to read through if you want to answer question on food biochemistry. Okay, so it's usually 1 mark for the statement, then 1 mark for the explanation, depending on how many marks is being allocated. Okay, if it's 25 marks, then 19 marks on the content. So maybe you can divide, but try to give as much as statement as possible, different points as possible. Okay, and make sure you explain the statement. Okay, just like this one. Okay, don't just write the calcium is important for development of bone and teeth. Then you can only get 1 mark. Okay, you have to explain how calcium help in the development of bone and teeth. Okay, and so on. Okay, if they ask you 5, then write 5 and explain the statement. Okay, so that's the question for food biochemistry. Okay. So next
Doktor,
one, yes.
yang for the case study punya soalan tu kan,
Mm-hmm.
yang five nutrients tu for the situation tu,
Mm-hmm.
do we have to make an introduction and conclusion, or just list down the 5 nutrients with the explanation and elaboration?
Still need intro and conclusion. So all essay questions still right. So you can give an introduction for what you are going to explain. So if the case study is saying about a patient with diabetes, so maybe you can explain what is diabetes. Okay, in the introduction.
Okay.
Okay. Okay, so the next question, just to show you how you answer if exams question ask you on comparison, or how would you differentiate. Okay, for this one, it's straightforward. The question asks you how group 1 and group 2 hormones differ in terms of their mechanism of action. Okay, what I'm worried is, I don't know because maybe you haven't revised yet, but most of you differentiate them according to their chemical structure. So you wrote group 1 is not sure. You compare, for example, peptides and amines lah. Okay, so the question is already stating here differentiate based on mechanism of action. So you need to describe the mechanism of action. Okay, for group 1 and group 2. So read the question carefully. Okay, so for a question like this, you can answer it in 2 different form. Okay, you can make a table like this. Okay, with the appropriate aspects. Okay, if you are comparing in terms of the receptor, so write in the aspect is receptor. Okay, and then solubility and then mechanism. So you need to write in the aspects. Okay, and whatever that you are going to write in the table, it doesn't have to be a really long full sentence, but don't make it like a hanging sentence lah. Try to make it proper. Okay, but for example, you are comparing in terms of the receptor, you can just write in, okay, intracellular receptor, okay, and then receptor at plasma membrane. Don't just write intracellular and then at plasma membrane. Okay, try to make it more proper, the sentence. Okay, or you can do it like this, or most of you did in the form of paragraph. Sini. Siap. Okay, or you can do it like this. Okay, one paragraph for introduction, and then one paragraph for group 1 hormone, and then the next paragraph is group 2 hormone, and then the conclusion paragraph. Okay, don't try not to combine all the answers in one paragraph. Okay, please make it easier for me to read. Okay, so this is how you answer for compare and contrast, or you are trying to explain two different things. Okay. Okay. So any question? I will upload the slides you can, so that you can refer later, also together with the recording.
So far now, madam.
Okay, hopefully you can study based on the tips that I've given. Okay, I have been giving a lot of hints already based on your practice questions. Okay, and tips on making the table. So I really hope you can revise, make preparations. Okay.
Yeah,
So
madam, noted.
if you later on you want to revise on the practice question and you haven't submit the GC yet, you can do so. You can still submit and just notify me if you want some feedbacks. Okay?
Yeah, madam.
All right, doctor.
Okay.